Hello, it's time to learn about another organelle now, the last organelle that we have to study. That's called the centrosome. Uh, I commonly call this organelle as the chief of partition. To partition is to divide. The word partition means to divide. Now, this organelle called the centrosome is involved in division of something. What does it divide? We'll see in just a little bit. But first of all, let us try to understand what kind of cells have them, or the distribution of centrosomes. Centrosomes are found only in animal cells. These organelles are found only in the animal cells. And where are they located inside the cell? They are found near the nucleus. So they are found here. Let me magnify it just a teeny weeny bit and see here. These are the centrosomes. They are found very close to the nucleus. Now let's demagnify and go back a little and also see centrosomes. So what do centrosomes look like? These both big pipe-like structures. You see, they are hollow on the center, but they are made up of straw-like structures. So both of them together make up the organelle called the centrosome. And each of this big tubular structure here, each of them, this one and this one, both of them together are called centrioles. So this is the first centriole and this is the second centriole, or you can call this the first, the second, doesn't matter. So there are two centrioles that make up the centrosome. Proceeding further, let us try to understand the structure of a centrosome. Now the organelles that you've studied so far, all the organelles that you've studied, the nucleus, the endoplasmic reticulum, the Golgi body, mitochondria, vacuoles, tiny little vesicles, or any other organelle that you've studied, even plastids, all of these organelles were or are surrounded by a membrane. And this membrane is just like the membrane of the cell itself. But what is unique about, not exactly unique, what is special about this organelle called the centrosome is that there is no membrane around it. So some organelles do not have membranes, have membranes. Now, pardon my sentence formation on the board. But anyways, let's come back. Now the organelles that don't have membranes, one of them is a centrosome, and there is another one which is a ribosome, the large molecule that makes proteins. This is also an organelle, but ribosome and centrosomes are called non-membrane bound organelles, which means organelles that do not have a membrane around them. Now let's proceed further and look into the details of the structure of this centrosome. What is it made up of? So we already know that they are made up of two centrioles, these two cylinders, which are again made up of straw-like structures. Yeah, so these are these straws, right? Hollow tiny tubes, right? So the structure of the centrosome, we have got a pair or two centrioles, and these two centrioles are embedded or they are found inside a jelly-like material, protein jelly. So I'm sure you have seen mixed fruit jam. So in a mixed fruit jam, we all have jam, right? And inside that you can have pieces of fruit. It's just like the same jam. Or you might have seen a cake with pieces or bits of fruits inside it. So it's just like that. We've got two centrioles embedded or found inside a protein jelly. So this is not liquid, but it's like jam, a jelly-like substance. Right? The jelly is made up of protein. The function of this jelly is to hold these two centrioles together and many other functions. But at the moment, just know this. Also, the centrosome is made up of these strings. You can see these bar-like structures. Bar mean rod-like structures, and these are not exactly rods. They are flexible and they can also grow. They are threads. These are protein threads. They are very long threads. Right? So this is the structure of the centrosome. What are these threads used for, and what these two centrioles do? We will try to understand them now in the functions. What is the function of the centrosome? So it seems like my computer has hung up because it is checking for some kind of update. But anyways, we'll try to continue by canceling this thing and coming back to the board. Now, technical issues, we can't help that out. So sometimes, we need to build up stories. We cannot stop the entire process. But unfortunately, it seems like the process has stopped. So we will have to open up the open board again. And I hope you stay with me because I'm not going to make the video again. Just bear with me a bit so that we can open up this board, and I hope that my content has been saved. Voila, it is saved. So let's go back. Thankfully, we are back. So we were studying centrosome, we were studying the distribution, animal cells, location inside the cell near the nucleus. We saw the structure of this particular organelle, the non-membrane bound organelle called the centrosome, the two centrioles, the protein jelly, the protein threads. Now, let us come to the function of this particular organelle. So first of all, let me also describe what kind of machine this is. Now, the two centrioles, they act like motors. I hope everybody knows what a motor is. A motor is a machine that can bring rotational motion into things. For example, your engine in the car acts like a motor, or we have got motors in toys that turn the wheels around. So these two centrioles act like motors, and these two motors have the function of pulling these protein threads. So what this centriole does, for example, this is the centriole, it has these protein threads. What the centriole can do, it can basically move the thread into itself, kind of like a loop. It doesn't loop exactly because the machine of the living world, the machines of the living world are different from the machines that we make. But these centrioles, they can pull these protein threads towards themselves. Now, why do they do that? It's something that we'll just see in a teeny weeny moment. And for that, we will have to watch a video. So let us watch the video first, and then we will see what is the function of the centrosome. Now, here is a wonderful video. I am going to give a running commentary, and I'm going to mute this video so that I can speak while I'm showing you this video, while you're watching this. So at this very moment, let me show you what this image has here. All of this red material is the DNA. It is the DNA of the cell. And this, where is the DNA found? Inside the nucleus. Now, when the cell is about to divide, the centrioles, I hope you remember centrioles now, when the cell is about, see, all cells must divide. See, that is the ultimate objective of life. All cells must divide. Let's write it down. So let us say this is the cell of our body. This is the nucleus. Inside the nucleus, we have the DNA. So this is our DNA. And we have all the other organelles in the cytoplasm, etc., etc. So the ultimate objective, what is the goal of all living organisms? The goal of all living organisms is to live forever. But can we live forever? Can I live for like 100 years? No, I cannot. So I will make babies. Just like plants make their own new babies by making seeds, the bacteria make babies, microorganisms, every living organism, most of the living organisms, they make babies. But how do they make them? They make them by dividing the cell. So this cell must divide and make two cells. And this is a universal thing. All cells divide to make two cells, whether bacteria, whether they are unicellular or multicellular, whether they are prokaryotes or eukaryotes, doesn't matter. Every cell has to divide. But if something has to divide, see, if this cell divides, it will make two smaller cells. If this cell divides, it will make again two smaller cells. If these cells divide, they'll make even smaller cells. If these cells divide, it will make two tiny cells. It doesn't happen so. So how do cells divide? The cells divide by, before division, the cells, they double up. They double up the amount of everything they have. So if they have cytoplasm, they will double up the amount of cytoplasm. If they have DNA, the DNA is also doubled up times 2. It is increased by 2 folds. It is increased in amount twice. So let us say this is the DNA, right? This contains all the information. Now the DNA is duplicated exactly, right? Because all the information that it has, it is copied by the cell exactly. So the DNA is doubled up. I'm using a red marker to show that this is the doubled up. This is the duplicate copy. This is the Xerox copy, the photocopy of this original DNA. Now the cell doubles up the DNA. And let me draw the DNA here. Now you see it is too much like biryani or like khichdi, right? It is kind of messy. Let me rub this a bit. I need to take this away. Let's rub this. Now the cell also divides all the organelles. The Golgi is divided, the mitochondria are divided, etc., etc. Everything is doubled up. Everything is doubled up. Now the cell is ready to divide. Now the cell divides by increasing in size. So size increases because cell membrane has also doubled up. And everything separates out to one side of the cell. Half of the content goes here, half of the content goes to the other side. Now when it comes to the division of the nucleus also starts to divide like this. You see? The nuclear membrane increases in size and now it has to divide. And these two DNA molecules must be separated out. So the DNA molecules are separated. Now the yellow strand must be separated from the red one, and something has to pull them apart. Something has to pull the yellow part, yellow DNA here, and something has to pull the red into the other side. And once this happens, what we get, once this happens, we get two cells. We get two cells. Oh, I've made a really large cell, a grown up one. So we get two cells. We get half a cell here like this, half a cell here like this. This is still dividing, okay? So that's why the membranes are still joined. And now we got two nuclei. One is here, one is here. Nuclei is plural of nucleus. One is nucleus, more than one nucleus are called nuclei. So one of the nucleus will now have this and the other has this. But what separates the DNA strands, the doubled up DNA from each other, that is the function of this organelle called centrosome. And centrosomes are only found in animals. Now how separation occurs in plants is something that we are not supposed to learn. But that is okay. We'll keep that for later. And now let us see how this happens. So we're going back to the movie now. And here we have that lovely, wonderful clip, courtesy Nikon microscopy. Nikon is a camera maker. They make fantastic cameras and microscopes. Anyways, coming back to the video. So here in the red, we see the DNA. You don't see any yellow DNA because I had the marker. The cell does not have any marker to show you two different types of DNA. So this is doubled up DNA. It has half the DNA is for one cell, half the DNA is for the other cell. And what you see here, this green blob, this green ball is one centriole, not the centrosome. This one centriole moves to the one side and the other centriole moves to the other side. These are like two motors, right? So we were talking about motors, right? So okay, here. So this goes to one side of the nucleus, this goes to the other side of the nucleus, when both of them carry with them a little jelly and protein threads. Now let's go back to the movie here and see. Now this is what is happening just before the cell is about to divide its DNA. So one centriole is here. Both of them together are called centrosome. They make up one organelle. Now this organelle has one centriole here and the other is here. And can you see these strings, the green ones? These are the protein threads. And they are going to split this DNA into two halves and then pack them into two cells. Now why am I talking? Let's watch the video. Whoa. Whoa, you see the splitting of the DNA? The partition? You see? This is slightly weird. But anyways, here you see three, but don't be bothered by it. See this one. Two centrioles, the protein threads, and the separated DNAs, and two new cells. You can watch this video over and over again if you wish to. Let's just finish this video and then we will say goodbye to centrosomes for this session. Cool. So, what is the function? Let us just finally put it into a statement. The function of the centrosome is to separate the DNA into two new nuclei or two nucleus of daughter cells. Daughter cells are the two new cells that are formed by the parent. Alright, so these are daughter cells when the animal cell divides. So I hope you've understood what centrosomes are. Thank you.